Good evening. Pansy the Answering Dog and I are here to go over the topic 4, body tissues and membranes, with you. And she has had her dinner, so she's in good shape. She was wagging her tail a moment ago. She was having apparently a happy dream because her eyes were closed. But she's awake now, so we're ready to go. Hope you're all set. And here we go. We're going to start with the body tissues and membranes. We're going to start with epithelial tissue. As always, I'd like to credit Professor Montgomery, Karen Montgomery, for all the support that she's given me, which I think in the long term pays off for you all. Epithelial tissue forms a barrier. It reduces water loss. It also reduces water gain. So if you're dehydrated and you put your hand in a bucket of water, you won't rehydrate. In fact, if you jump into a swimming pool, you won't rehydrate as long as you don't drink the water. I would recommend that you not do that. Not jump in the swimming pool. You can jump in the swimming pool, just don't drink the swimming pool water. See, Pansy likes to jump in swimming pools. It prevents abrasion. So when you rake leaves, for example, if you do that in the fall and you don't wear gloves, the epithelial tissue will keep the underlying tissues from being damaged by the raking action. Also, if you gradually use your hands for something like raking, you'll build up calluses, and the calluses will further help prevent abrasion. Although a lot of people are not, or they're put off by calluses. Calluses are actually a very useful part of your skin. It protects against UV light. Now, UV light is kind of a mixed bag. We need UV light in order to synthesize vitamin D, but too much UV light is bad for your for your genetic material. UV light is a high energy electromagnetic radiation, and it can damage your DNA. And eventually, if you have enough damage, it tends to be cumulative, and you'll end up with skin cancer. Anytime we have exchange at a surface, or nearly any time, it will involve epithelial tissue. An exchange includes 3 categories: secretion, absorption, and excretion. I didn't put excretion in here, but because that's sort of limited, but there are a couple of organs involved in excretion. And in any time you have materials leaving your tissues or coming into your tissues, with the exception of the synovial membranes in your joints, it will involve epithelial tissue. The epithelial tissue is going to cover and line your body, the ducts and the vessels, things of that nature, your gastrointestinal tract, aorta, anything like that. It prevents bacterial invasion. This is actually pretty important. We get to take this sort of for granted, but if you get cracks in your skin, for example, and you aren't careful, you can end up with those cracks becoming infected. Bacteria can get into your tissues and damage your tissues. If your skin is healthy, that pretty much doesn't happen. Part of some epithelial tissues would be an organelle called cilia, or cilium would be singular, cilia is plural. The cilia are extensions of the cells, and they push material out of our respiratory passages especially, so that helps us clean the air before it goes into our lungs, and that's very important. It's not the only thing that helps clean the air, but it participates. There are several different kinds of epithelial tissue. There is a simple epithelium where you just have a single layer of cells, and then we have stratified epithelial tissues, and those have at least 2 layers of cells. It could be 2 layers or more. There are 3 kinds of cells if we categorize them by shape. We have squamous epithelial tissue, or squamous epithelial cells, and those epithelial cells are going to look a lot like a pancake. They're flattened. We have cuboidal epithelial cells, and even Pansy knows what shape they are, right, Pans? What are they? Right, cubes. And then we have columnar. Columnar epithelial cells look like, right, columns. And columnar cells are actually named for a famous Spanish anatomist and then later explorer of the world, Christopher Columnar. Oh my God. Pansy's rolling her eyes at that one. There are going to be two kinds of specialized epithelial tissues, transitional epithelium and pseudostratified columnar epithelium. Pseudo means what, Pans? Right, false. So pseudostratified columnar means that it looks like it's a stratified columnar tissue, but in fact it's not. It has to do with the shape of the cells. And we'll actually see some drawings of pseudostratified columnar epithelial tissue. So where do we find some of these tissues? The simple squamous epithelium form capillaries. Capillaries are the functional units of our circulatory system in terms of exchange between the blood and the cells surrounding the capillaries. They're really, really important, and the fact that there's only one layer is also important because it facilitates the exchange. It makes there less obstacles for the materials to leave the blood or go into the blood. We have the stratified, we mentioned that. And then there is a special kind of stratified squamous, non-keratinized epithelium. Those tend to be found in moist surfaces, say in your mouth, for example. And then we have stratified squamous epithelium that is keratinized, and there's a protein, keratin, that reinforces the cells and makes them a little bit tougher and less permeable. Here's a model of a squamous epithelial cell, and if you remember I said they look like pancakes, but if you look at this cell it looks more like a cucumber. And the reason is you're looking at the side of the cell. So the cell has been cut open. The dark part is the membrane. This lighter portion is the cytoplasm or cytosol. We'll be talking about that. And then the thing that looks like a blue M&M's chocolate covered peanut is actually the nucleus. And then this is a stand for the cell. And we have stands because this is a model. Okay. Here we have the surface of the squamous cells. They look a little bit like a mosaic. And in fact, you can see an interesting characteristic of the squamous cells in this drawing, or in this model. You see that the cells run right into each other, almost as though you had poured pancake syrup into a pan and they didn't cook fast enough and ran into each other and sort of made one big huge pancake. That's sort of what we have with epithelial cells. Usually there is no or very little intercellular matrix between the cells. Here are some epithelial cells of an individual that have been dyed to make the organelles a little bit more obvious. And in this cell right here, you can see that it looks like the nucleus is a figure 8. Actually what's going on is this cell is undergoing mitosis, cellular division, and before very long there's going to be a constriction of the cell right about here, and you're going to end up with two new cells, two daughter cells, where we originally had just one cell. Cuboidal epithelium, as Pansy so brilliantly pointed out, is going to be sort of cubic shaped. And there are a couple of different kinds of cuboidal epithelium. We have the simple cuboidal epithelium. These usually secrete, or if in some cases they absorb, and if they do absorb materials, they usually have microvilli, which are little projections off the surface of the cell. These microvilli serve to increase the surface area to volume ratio, which is a very important principle all throughout anatomy and physiology. Anytime you have exchange with the surroundings, whether things are coming in or going out, it is helpful to have extra surface area, and these microvilli do that for us. Sometimes people think microvilli are the same thing as cilia. They are not. So the microvilli don't push. They just help increase surface area. Cilia would push. And then we have stratified cuboidal epithelium, and these tend to line the ducts of larger glands, such as the liver or the pancreas. Here is a model of cuboidal cells. We see they have the same features found in the squamous cells, just with a different shape. So we have the membrane here on the outside, the dark, and then the cytoplasm or cytosol, which is kind of this yellowish greenish stuff. We have the M&M chocolate covered peanut, but that's not really what it is, right? That's the nucleus. And then because it's a model, we have a stand. And here's the surface of the cuboidal epithelial cells, and something might already be occurring to you. Again, we don't have any, or if we do, very little intercellular matrix, but if you just looked at the surface of these cells, it would be hard for you to determine whether you're looking at cuboidal cells or squamous cells. Columnar cells are going to look like columns. Simple columnar epithelium often create mucal secretions. They're a special kind of cell called a goblet cell that produces the mucus. If the columnar cells are absorptive, as they would be, for example, in the small intestine, they would have the microvilli we mentioned before, which increases surface area. And then in some places, these columnar cells will have cilia. And a good example of simple columnar cells with cilia would be in the uterine tubes. Uterine tubes, by the way, have a couple of other names. Sometimes they're called fallopian tubes. Sometimes they're called oviducts. So basically the same. And then we have the stratified columnar epithelium. We find those in a couple of places inside the male urethra, inside the pharynx. Pharynx is between your oral cavity and your esophagus and trachea. And then we have the pseudostratified columnar epithelium, and you recall I mentioned pseudo means false, so this appears to be stratified, but it's not actually. And basically, the reason this impression is given when you look at these cells is that they look kind of like a candle. They tend to be narrow at one end and wider at the other end, and the wide end is usually where you'd find the nucleus. The places that we're going to find pseudostratified columnar epithelial, cuboidal, no, these are columnar cells. That's a mistake in this word right here should be columnar. They're going to be in the reproductive tract. Some are in the respiratory tract. And as I mentioned before, some of these cells are called goblet cells. They produce mucus. Mucus in the respiratory tract has a bunch of functions. Warms and moistens the air, filters the air, and is bactericidal. And then some of these cells are going to have cilia, and the cilia move the mucus. We'll see a little bit of example of cilia. I guess we already did that. We saw those back when we did organelles in quiz 3. Again, there's a mistake here. Dr. J is dropping the ball. That should be columnar. We also find the pseudostratified columnar epithelial tissue in the respiratory tract. Well, actually, we already mentioned that. So here's what the mucus does. I guess I already told you. It traps debris. It warms and moistens the air. And then the cilia push the mucus out of the respiratory tract. Here is a model of a section through columnar epithelial cells. Same features that we saw in cuboidal and squamosal. We have the cell membrane, the cytoplasm or cytosol, we have the nucleus, and because it's a model, we have a stand. And then from the surface, again, you can see that these look virtually identical to cuboidal cells from the surface. There's very little or no intercellular matrix. Cells run into each other sort of like pancakes. Here are some pseudostratified columnar epithelial cells that have cilia. And remember I told you they look a little bit like a candle. So here's the narrow end of this cell, here's the narrow end of this cell, and then the nucleus tends to be closer to the end that's thick. Then we have transitional epithelium. Transitional epithelium changes in response to tension, and we're going to find that it lines the urinary bladder, it lines the ureters. Ureters lead to the urinary bladder from the kidneys. It lines the urethra, and the urethra is going to carry urine from the urinary bladder out to the outside. Cells that are transitional have the ability to change shape depending on how much tension they're under. Usually, if they don't have any tension, they're cuboidal in shape. But if you put them under tension, that is, if you stretch them and you pull on them, they become flattened like squamosal cells. And then, if you release the tension, they tend to go back toward the cuboidal shape again. Okay, epithelial tissue, as I mentioned earlier, has little or no intercellular matrix. So if you had a wall, as we do in school, that's made out of cement blocks, and you had mortar between the blocks, if the cement blocks were the cells, there would be no mortar, or if there were any, it would be very little, very minimal. We already mentioned cilia. Some of the epithelial cells have those, not all. Epithelial cells have cilia. Cilia are an interesting organelle. We talked about those back in the Quiz 3 materials. Cilia are a non-membranous organelle used to propel material adjacent to the cell in humans, whereas in single-celled organisms, the cilia would propel the cell themselves. Okay, we find them in 3 places: respiratory passages, the uterine tubes, oviducts or fallopian tubes, and the ventricles of the brain. In all of these places, they move the material adjacent to the cell because the cells are fixed. I should mention that cigarette smoke has a negative impact on the cilia. It makes them stop working. And so if you're smoking cigarettes or if you're inhaling cigarette smoke, your cilia and your respiratory passages don't work. They don't remove all the garbage, and that material will go down into your lungs normally because of gravity. This is not desirable. The good news is that if you stop smoking for a while, your cilia can recover. Here is an individual cilium. Although there is a plasma membrane around the outside of the cilium, it doesn't completely encapsulate it, so they do qualify as a non-membranous organelle. And then inside there are going to be 9 pairs, microtubule doublets. And those microtubules are responsible for the ability of the cilia to push. And the really cool thing is that they all get coordinated, so they all push in the same direction. So cilia move the material adjacent to the cell they're part of in humans because the cells are fixed in position. We're done with cilia now. We'll talk about some of the other places we find epithelial tissue. You remember that I said almost always if we have exchange, it's going to go through epithelial cells. Epithelial cells are usually part of glands that secrete because the epithelial cells make the secretions. Some epithelial cells have sensory receptors, and these oftentimes, not always, but often are sensitive to compression, because when you compress the cell, it changes its shape. Usually it stretches the membrane and makes the membrane leaky with respect to the ions that cause impulses to arise. Okay. This is a really cool and somewhat unusual characteristic of epithelial tissue. Epithelial tissues usually, and we'll see that there is an exception in the lungs, but usually they're capable of significant regeneration. This is hugely beneficial since they are the cells that are adjacent to your environment. They are the cells that are most likely to be damaged by interaction with your environment, and the fact that they can regenerate themselves, replace themselves, is important because remember they're a barrier, and if that barrier goes away, you're at risk for infections and things of that nature. If you have normal circulation, The regeneration of these cells should be rapid. So, for example, if you get a paper cut, something of that sort, it should heal fairly quickly, usually within a week. Obviously, if you have some really serious cut, like with a chainsaw or something, it can take a lot longer. But if it's just a simple cut, it should heal. And I would mention that if you have a simple cut and it doesn't heal, you should be concerned. You should probably see a doctor about that. Could indicate that you have poor blood circulation in the area, but it certainly would merit looking into. Normally these epithelial cells are attached to a basement membrane. This is a non-living layer made up of carbohydrates and proteins that's deep to the epithelial cells. Now they are non-living, they are organic, so your cells produce that material, but they're not a living tissue. In most cases, especially at the surface of your body, the epithelial tissue doesn't have any vascular supply of its own. And you might think, wow, how can they survive? The reason they can survive is because they're fairly low maintenance. They don't require a lot of oxygen, they don't require a lot of glucose, and they don't produce a lot of waste products. And they get what they need from the vessels and adjacent areas in connective tissue typically. Organs that are mostly epithelial tissue. I think it's useful, I think if you remember we talked about organs often being primarily one kind of tissue. I think it's useful if you give examples of that so you can kind of see how that goes. The skin is mostly epithelial tissue. The pleura inside the thoracic cavity and the peritoneum inside the abdomen are principally epithelial tissue. And then the lungs, the functional portion of the lungs, the alveoli, are sacs made out of epithelial cells. And that brings us to connective tissue. So connective tissue, the name sort of gives you a hint as to one of its functions, right? What would that be, Pans? Right, connect. The connective tissues hold things together. Okay. Connective tissues can protect. They can form bumpers or cushions, if you will, to help reduce damage to underlying tissues. And they, in some cases, are found in between organs or tissues, and their presence reduces abrasion of those tissues. They can support our weight against gravity, these connective tissues, and we'll talk about specific organs that are primarily connective tissue that do support you against gravity. The bones, right? Which is one of Pansy's favorite toys, a bone. They insulate, and I think we'll probably expand a little bit on this. There are two kinds of insulation. There's insulation against thermal exchange. Heat loss during the winter is reduced if you have adipose tissue, which is a connective tissue. Whales and other marine mammals depend on this insulation to keep from freezing to death. But we also have insulation around our nerves. That prevents the impulses in the nerves from spreading into the surrounding tissues. Bulk flow is, well, there are several examples actually of bulk flow in our body. The one that involves connective tissue is the circulatory system. Blood is a connective tissue, and it is what is pumped throughout the body. Immunity against infections or toxins. Most of our immunity is brought about because of the activity of white blood cells, either directly or indirectly, and the white blood cells are part of the blood, and the blood is a connective tissue. And then storing excess energy. We'll actually expand on this a little bit. There's kind of an interesting twist to this that we'll mention when we compare animals and plants with respect to how they store excess energy. As storing excess energy probably doesn't seem like a big deal to you, but actually it's very important. Most of the animals in the world, with the exception of human beings in developed countries, or at least most human beings in developed countries, most animals in the world spend a large fraction of their life just trying to find enough energy sources to survive. Energy sources are food, right? And if they are fortunate enough to find a surplus of food, they often eat that extra food and then store in their tissues the extra energy that they have consumed. And we will talk about the advantage of using connective tissue for that. One of the characteristics of connective tissue is that it has a lot of intercellular matrix and not very many cells. Not having very many cells is good news and bad news. Remember, I've said that almost any time there's good news, there's bad news. The good news about not having many cells is that it's fairly low maintenance tissue, so it doesn't require a lot of oxygen, it doesn't produce a lot of waste products, it doesn't require a lot of glucose. That's the good news. Bad news is that if you damage connective tissue, it can take a significant amount of time to heal. That doesn't mean that it won't heal. They heal. They just take a lot of time, unlike the epithelial tissues we talked about earlier. The matrix, the intercellular matrix between the cells, is organic material. It usually includes fibers, protein fibers, that make it strong. Although, honestly, in some cases, the tissue is not very strong. A good example would be loose connective tissue like the fascia. The connective tissue, unlike other kinds of tissues, can exist either in a solid state or in a liquid state. And the most important of the connective tissues, one of the most important ones, is blood, and that obviously is a liquid, which makes it easy to pump and makes it really useful for delivering materials because of the high concentrations of materials within the liquid. The matrix of any given connective tissue is going to affect the properties of that individual tissue, and we will mention some examples here. So one of the things that makes up the matrix, we talked about fibers, is collagen fibers. Collagen is a strong protein. It's flexible, so it allows things like bones, for example. Even though they're fairly rigid, bones can actually bend a little bit, and that ability helps them do their job. There are also another kind of fibers, elastic fibers. We won't talk about elastic fibers as much, but there is a protein called elastin, and this is going to stretch a little bit more easily than collagen would. And then there's a third kind of fiber, and those are the reticular fibers. These are found in cartilage, cartilaginous fibers, and they help create strong networks within the tissue. Now, in terms of types of fibrous connective tissue, we have loose connective tissue or areola connective tissue, and this is not strong. This would be, an example would be the fascia, and it tends to occur between other tissues or between organs. If you were, and some of you may have seen this, if you take a piece of chicken before it's cooked and you pull the skin away from the muscle, you'll see something in there that looks a lot like cotton candy or like a spider web. It's not very strong. It can be easily torn. That fascia is found in between all the layers of the body, or muscle layers of your body. So you have superficial fascia between the skin and the muscles, and then deep fascia between the muscle layers, and it can be torn pretty easily. Most cells are fibroblasts in these loose connective tissue, and they are able to produce those fibers outside of the cells. Remember that red letters indicate function in these PowerPoints. The cells in the loose connective tissue are separated by fibers of both collagen and elastic, and the fibers are going to be important for stabilization of the matrix, so it doesn't all fall apart. It tends to make it cohesive, and it also is able to stretch and then return to its original length after stretching. There are some limitations to this. So, for example, we find loose connective tissue in the mammary glands, and if the mammary glands stretch too much, these fibers are not capable of returning to their original shape. Adipose tissue is an example of loose connective tissue. Adipose tissue, when we were in nursery school, we used to call what, pants? Right, fat. And the fibroblasts of the adipose tissue actually store lipid compounds, mostly neutral fats, and there isn't a whole lot of material between the cells. The cells make up most of the tissue. Adipose tissue is really good at a couple of things. One is both thermal and electrical insulation. It's an insulator, and it cushions us. So if you have layers of adipose tissue, it can prevent physical harm to the deep layers when you receive physical impact. Excuse me. As I think I mentioned earlier, adipose tissue is the primary way we store excess energy. Now, plants don't store excess energy as lipids usually. They store most of their excess energy as carbohydrate, and specifically starch. So what would be the advantage of storing lipids instead of starch, Pansy? You know this. Right. One is that fat is lightweight. And if any of you ever counted calories, you know that if you buy an article of food at the supermarket or wherever, and it is made out of a pound of cornstarch or starchy materials, it's not going to have as many calories as if it's made out of a fatty material. And in fact, you see here, the ratio is about 2 to 1. You get about twice as many calories in a pound of fat as you do in a pound of starch. So if we stored 1,000 calories, let's say, in tissue that had starch, that tissue would weigh about twice as much as if we stored the same number of calories in a tissue using fat. The other thing is, right, flexibility. Okay, and adipose tissue is more flexible than starch. So the obvious question then, Pansy, is how do we benefit from these two attributes of storing fat? And that would be, right, speed. This is one of Pansy's favorite things, because when she goes outside and she sees some chipmunks or something, or squirrels, she tries to catch them. The good news for the squirrels and chipmunks is that they're faster than Pansy, so they never get caught. The good news for Pansy is I feed her, so she doesn't have to depend on eating squirrels and chipmunks. We have dense connective tissue. Dense connective tissue is very strong. It has thick bundles of collagen fiber in it to give it that strength. And there are a couple of different kinds. There's dense regular connective tissue. In these cases, the fibers are, as for example in tendons, ligaments. Ligaments are very similar to tendons, except they exist between bones. Tendons usually exist between muscles and bones, or muscles and other muscles. And aponeuroses. Aponeurosis is a special case of tendons. It's a broad, flat tendon. Okay. We also have dense, irregular connective tissue. And in this case, the fibers don't all line up and run parallel. They run in different directions. And we're going to find some of this dense, irregular connective tissue deep in the skin, in the dermis. We also find it in joint capsules. Joint capsules have this dense, irregular connective tissue, and because of that, because they're strong, they give support to the joints and help reduce the risk of damaging joints. They stabilize your joints. Okay. Reticular connective tissue is another type. Here, the fibroblasts are called reticular cells, and the only thing in between the cells are the fibers, the reticular fibers. This is also sometimes called lymphatic tissue, and we have lymphatic tissue in another number of places. We find them in the lymph nodes, in the spleen, thymus gland, which is a part of your lymphatic system, and red bone marrow. And this is an important characteristic of all of those lymphatic tissues. The reticular connective tissue is going to be an important part of our immune system, and this is because it stores and produces white blood cells, and one of the type that's a blood cell that it is especially good with storing and then producing as needed are T lymphocytes, or B lymphocytes for that matter. The B lymphocytes produce antibodies, and they stay in place, but they produce these proteins that are released into the blood, and they help us deal with certain kinds of infections and toxins. And then we have the T lymphocytes, and the T lymphocytes are responsible for cell-mediated immunity, and they actually travel to the site of an infection to help us deal with those kinds of things. So all the blood cells, red blood cells, white blood cells, and platelets originate from red bone marrow. Cartilage. Cartilage is similar to bone. In fact, bones all originally were cartilage. And if you look back in the vertebrate lineage in terms of evolution, the earliest vertebrates didn't have any bone. They had exclusively cartilage. The cells of cartilage are called chondrocytes. You know, chondra means cartilage, right, or granule. They live in lacuna, and the lacunae are like little caves within the matrix. And the matrix in a cartilage is flexible, unlike bone, which would be rigid. There are a couple of different kinds of cells. The chondroblasts are immature cells. They cause the cartilage to grow. And then the cartilage is based on, the classification of the cartilage is going to be based on the kinds of fibers that are in the matrix, and we have really three kinds of cartilage. So here are the three kinds. Hyaline cartilage, we find this at the end of long bones where they articulate with other bones. It's kind of rubbery. It's usually smooth, fairly dense, and it's a shock absorber, but it also, because it's smooth and because it gets lubricated with synovial fluids, it has very little friction between that and the adjacent bone. The elastic cartilage is going to be a little bit stretchy, and fibrocartilage is not stretchy very much at all. It's rather strong, tough, and stabilizes the objects that it is attached to. So the hyaline cartilage, we'll go into a little bit of detail about each one. Hyaline cartilage, most common, strong, durable, and flexible. And I mentioned the end of the long bones because of its importance in the articulation of those bones when we move, but you also have a scaffolding in your nose other than the bone, and that scaffolding is made out of hyaline cartilage. And then the tracheal cartilage, the tracheal rings, are hyaline cartilage. So they're flexible, but still offer some rigidity. Then we have, I mentioned that bones start out as cartilage. The fetal skeleton is actually pretty much all hyaline cartilage, but eventually that cartilage becomes bone tissue. We have elastic cartilage, a lot of elastic fibers, going to be kind of stretchy yet and strong. And it's found in your outer ear. It is more flexible than hyaline cartilage. As I said, there aren't going to be as many examples of elastic cartilage. And then we have fibrocartilage, and remember I mentioned it's kind of strong and not particularly stretchy. This is because of the collagen fibers within the fibrocartilage, and the fibrocartilage is going to absorb shock. It also is going to help cope with tension and pressure when you're pulling on one part of the body or another, that these parts are connected by fibrocartilage. We find fibrocartilage in the intervertebral discs and also in the meniscal cartilages of the knee. There are two meniscal cartilages in your knee. Okay, bones are next. Bones have three components. We'll spend a lot more time on bones. It's one of the upcoming lecture topics, actually. But one of the most dominant component of bone tissue is hydroxyapatite. Hydroxyapatite is an insoluble salt. It's inorganic, so it doesn't involve carbon or hydrogen, and it's like a rock, which was actually a pretty good song by Bob Seger. We also have the bone collagen, which is the protein fiber material that runs within the matrix, and this acts sort of like rebar in cement construction. So some of you have probably seen cement structures being built, and they don't just pour cement, they put in these metal mesh or rods to help make it stronger. And then there are going to be the cells, and the cells are capable of increasing the amount of material with growth, or there are other cells that actually erode material. Bone is very rigid material. Actually, this is kind of an interesting fact for you. Bone is stronger per pound than steel. Now, obviously, your bones don't weigh as much as they would if they were steel. So this is a trade-off, right? It is lighter, and light is good for speed. And bones are really strong, and that's good also. It's not as strong. If you had a femur made out of steel, it would be stronger than the femur that you have, but the downside would be that it would be a lot more weight. Remember, low weight is good for speed. Okay, there are going to be 4 kinds of cells. There are these osteoprogenitor cells. They're not really specialized yet. They're sort of like stem cells. Find them in the periosteum and in the endosteum. Endosteum is inside of the hollow space of a bone and the central canal. We have osteoblasts. Osteoblasts actually make new bone tissue. This happens in a couple of situations. One is when you get taller, the bone grows longer and the bone gets bigger in diameter. This is because of osteoblasts. But also they work when you break a bone and you have to form new bone tissue to get the bone to heal. Osteoblasts do that. And then we have osteocytes. When an osteoblast gets incorporated into the matrix of the bone, it's called an osteocyte. And then we have osteoclasts. And osteoclasts are sort of the opposite end of the spectrum from the osteoblasts. Instead of making new bone tissue, osteoclasts erode bone tissue. This is really important in 2 instances. One is as you grow, the central canal, the medullary canal or marrow cavity of your long bones gets bigger, and so the bones become more hollow. You have to have some cells that can erode the bone tissue while the osteoblasts are adding to the bone tissue. It's also important when you break a bone. These osteoclasts can help reconfigure the bone tissue. Possible that the osteoclasts come from monocytes. So the obvious question is, well, how can a cell erode bone tissue? I mean, bone is an insoluble inorganic salt, so how do you get that to disappear? The osteoclasts use an acid based on carbonic or carbon dioxide, and that acid is used to erode the bone, sort of the same way some of you have probably seen those limestone caverns. That's all possible because of this reaction of carbon dioxide and water forming a weak acid, carbonic acid, and then acids dissolve bone tissue. Compact bone. Compact bone is going to have a Haversian canal system, or osteons. There's going to be a central canal where the blood and nerves run, and then there are rings called lamella or lamellae, which include the cells, and they are arranged around the cavity with the blood going through it. Okay, we have canaliculi. Canaliculus is a little tube, right? Iculus is diminutive, and canal is a tube. These are teeny tubes, which almost rhymes there, right? These are teeny tubes, or tiny tubes, depending on how you want to call it, that basically extend from the lacuna where the cell is down to the Haversian canal where the blood is. And then the cell has an extension of itself that goes down through these tubes to get close to the blood. So here we have a fairly reasonable drawing. The Haversian canal would be where the blood would pass through. These little red things are the osteocytes, and they're caught within the matrix. And then these little lines are actually tiny little canals that the cells extend themselves through down toward where the blood is. Spongy bone is a special kind of bone. It's named for a famous anatomist who later became a TV personality, Spongy Bob. We find it in a couple of places. It's going to be at the end of long bones. Spongy bone is where blood cells are formed. And there are trabeculi. Now, trabeculi means beam, B-E-A-M. So these are bony bars or beams and plates within the spongy bone. And then there are these spaces between them. Even though it doesn't look very strong, the trabeculi are actually fairly strong, which is good because it stabilizes the bone. And the blood cells are going to be formed in the red bone marrow of long bones that are found within the body. When you're a newborn, pretty much all your bones form red blood cells, but as you grow older, the number of places that form red blood cells reduces. So here we have a cutaway. This is a femur, and if you expand this picture, you can see all these spaces. Here's another one. So we have this outer layer of dense bone, and then the inner layer is the spongy bone. The dense bone or compact bone is very hard. It's like a rock. Here's a pretty good drawing of what the trabeculi are like. So there's going to be bone marrow and blood vessels in some of these spaces, and then these little columns of hydroxyapatite are the bone matrix itself. Blood is kind of an interesting connective tissue. It's the only major connective tissue in our body that's a liquid, and that liquid nature actually turns out to be kind of important. It has plasma. The plasma is the interstitial fluid of the blood. This occurs between the bones, or between the cells rather. And then we have the formed elements. The formed elements exist in three cell types. We have red blood cells. They are also called erythrocytes, and you know from your zoological roots that erythro means red. They are important for transporting oxygen and carbon dioxide in the blood, and they also help buffer the pH of the blood, because the hemoglobin that they contain can act as a buffer. White blood cells are sometimes called leukocytes, and they're named after a famous anatomist who later became a very important character in the Star Wars, and that would be Leukocyte Skywalker. Some of you probably heard of him. There are 5 different kinds of leukocytes. We'll talk a little bit more about those. And then we have platelets. The leukocytes are involved in immune responses. Platelets are sometimes called thrombocytes, and they are important for blood clotting. So the red blood cells, here are our functions. Remember, red writing usually indicates functions. They carry oxygen, carbon dioxide, and they adjust the pH. And they all, the molecule that they have in them that makes this all possible is the hemoglobin. Should also mention red blood cells, as we'll see, are kind of a special shape, and they don't have any nucleus. Not having a nucleus is good news and bad news. And the good news that is that there's more room for hemoglobin, and the bad news is that they can't replace themselves as they get old. So you have to get new red blood cells from the bone marrow. The white blood cells are involved with immunity. That's really a big deal. I mentioned there are 5 different kinds. We'll talk a little bit more about those in a moment. And then the platelets or thrombocytes are important for blood clotting. Now they don't actually form the clot, but they are involved in the formation of the clot. There are some proteins in your blood that form the clot, and then these proteins interact with the platelets, and the platelets actually squeeze fluid out of the clot as the clot forms. Platelets appear to be fragments of large cells that are in the bone marrow called megakaryocytes, and they do not have a nucleus. So because they don't have a nucleus, that means they can't divide either. So as you have platelets damaged or destroyed, you have to get new ones from your bone marrow. Here's a pretty good photograph from a microscope of red blood cells, and we notice a couple things. First of all, they're biconcave discs. That means that they're sort of like a cream saver. They're thicker around the outside and kind of thin in the middle. They're extremely small. Both of these characteristics, the biconcave disc shape and the fact that they're very small, increases something. So what does it affect? Pansy knows this answer. Oh. Right, surface area to volume ratio. Isn't she wicked smart? She always amazes me. So surface area to volume ratio is very important anytime we have exchange, and that's what the red blood cells do. They pick up oxygen in the lungs and drop off CO2, and then they drop off oxygen in the tissues and pick up the CO2. They sort of reverse their role in that way, and I think we already saw a thing on diffusion. Remember, things go from a high concentration to a low. So if the oxygen is in high concentration outside the cell, as it is in the plasma in the lungs, then the oxygen goes into the cell, and the CO2 is high in the cells in the lungs, so it leaves the cell and goes into the plasma and then eventually the air. And then it's just the opposite when you get to the tissues. So the oxygen leaves the cell and the CO2 goes in. Here's a neutrophil. This one has a number of nuclei, 4 in particularly, and neutrophils are one of 5 different kinds of white blood cells. Remember that they're involved in your immune system. The 5 kinds are neutrophils, eosinophils, monocytes, lymphocytes, and basophils. And we'll talk more about those when we get to blood. Also, you see that outside of this, there are these kind of dull pinkish colored things that are kind of light colored in the middle. Those are red blood cells, and the light color in the middle indicates that the middle is thinner than the outsides. That's why it's like that. The ratio of neutrophils to red blood cells in a healthy person is about 1 to 1,000. 1 white blood cell for every 1,000 reds. Here are some platelets. And remember, we said that platelets are important for clotting, and you can see they're really, really small. They're just fragments of the megakaryocytes. And then again, we see these red blood cells here with a light center, indicating that they are thinner in the middle and thicker around the edge. There are some cells called stem cells that live in the red bone marrow. We've talked about stem cells before. Remember, in topic 3 we mentioned that stem cells are cells that sort of stay in an immature state and are capable of reproducing and differentiating as they do. They sort of stay as an undifferentiated cell to begin with, and these are the cells that give rise to the blood cells. They have a very high division rate. That's really important because that means that they can form new cells quickly, and we need that because we lose red blood cells and white blood cells and platelets every day, and most of them are being replaced from the bone marrow. Then we have the plasma. We already talked about the plasma. That's the material surrounding the cells. That's the interstitial fluid. In this case, it's the intercellular matrix. And the plasma, by and large, is not really formed by the cells. And what we find in the plasma is actually controlled and managed, added to, and taken away from by the various organs throughout the body. Okay. Organs that are mostly connective tissue, I think the bones are really the best example. I can't think of a lot of other organs that I would say are connective tissue, but bones definitely are. Which brings us to muscular tissue. And muscular tissue has cells that are able to contract. And we use these contractions basically for 4 functions. I think we've talked about these perhaps before, maybe not. So just to be sure, have to bear with me. I have so many classes, sometimes it's hard to remember exactly what I told everybody. But they are involved with Pansy, right, creating pressure gradients. And we'll expand on this a little bit. Another thing, right, thermoregulation. Thermoregulation is good news and bad news. We'll expand on that a little bit too. Another thing, right, support against gravity. She's really good tonight. And then the last one, I'm going to give her a break because she's looking like she's fatigued. Movement. The contractions of muscular tissue, specifically skeletal muscle tissue, is going to be involved with movement. And I'm sure you remember, I think we talked about this, there are 3 kinds of cells in muscular tissue. In some of the areas we have transition zones from one kind of cell to another. Good example would be the superior end of the esophagus. The superior end of the esophagus is more like a skeletal muscle than the other 2 kinds of muscle. And then as you progress toward the stomach, it transitions over from skeletal to smooth muscle. Same thing happens at the end of the large intestine where we go the other way, from smooth muscle back to skeletal. In the heart and in the aorta, we find that the very beginning of the aorta is more like cardiac muscle tissue than smooth muscle tissue. But as we go away from the heart, it transitions over into pretty much the characteristics we would expect from smooth muscle. There's some bad news that I can't think of any good news as a counterbalance, and that is that cell division is pretty much complete at birth. So muscle tissue is not very good at repairing itself. If you get injured, usually and you kill muscle cells, you don't get new muscle cells to replace them. There is some evidence that occasionally muscle cells do divide, but for the most part they don't. Okay. The cells themselves are referred to as muscle fiber, and by the way, there is going to be a separate quiz topic just specifically on muscles that will be coming up. And they contain these protein fibers called myosin fibers, and the myosin fibers interact with each other, and that results in movement of the cell. Basically, the cell gets shorter, typically. And we mentioned there are 3 kinds of muscle tissue. So we have skeletal muscle tissue, sometimes called striated or voluntary. It's called striated because it has stripes. If you look at it under a microscope, that's really a physiology thing that we'll talk about when we get to that muscle quiz. It's called voluntary because for the most part, if you want to contract a skeletal muscle, you can do that. And it's called skeletal because it usually is attached to the bones. We have smooth muscle. Smooth muscle has 2 other possible names. Sometimes it's called involuntary muscle. Sometimes it's called visceral muscle. It does not have the stripes that the skeletal muscle has. It's called involuntary because typically you can't contract a smooth muscle because you decide you want to. And it's called visceral because it frequently is associated with visceral organs. And then we have the cardiac muscle tissue. And I didn't give any alternative names for it. Cardiac muscle tissue could be considered involuntary. Again, you can't really contract cardiac muscle tissue because you want to. And I should mention too that some books lump things together, so they only end up with 2 kinds of muscle tissue, and the cardiac muscle tissue, they slide into one of the other 2 groups. It really has characteristics of both. There are some characteristics of cardiac muscle tissue that is very similar to skeletal, and other characteristics that are very similar to smooth. So I think it's reasonable to categorize it as its own special type. Skeletal muscle cells, what do they do for us? Well, first of all, as I already mentioned, they're usually attached to bones by tendons, and they perform all 4 functions. When you walk, you use skeletal muscle tissue to do that. When you stand up and don't fall down, you use skeletal muscle tissue to hold the bones where they belong. When you're cold, you shiver. That gives off heat. About a little more than half of the energy that's released when muscle cells contract, more than half of the energy comes off as heat. And so if you're cold and you shiver, those muscle contractions warm you up. And that's good news. Bad news is if you're warm and you use muscles, you warm up more, and if you get too warm, it can actually cause you to be dead. And then the pressure gradients. We talked about that in class. Remember the skeletal muscle pump working in conjunction with the one-way semilunar valves? That's how we get blood to come back from our extremities, but not just blood, lymph as well. So skeletal muscles can create pressure and move materials because of that. We call them voluntary, but actually sometimes they're involuntary. When you have reflexes, you have skeletal muscle contractions that you're not actually consciously controlling. That's good news and bad news. Reflexes are fast, that's good news, but sometimes you make inappropriate movement, and that's not good news. The cells tend to be very long, and in fact the really cool thing about skeletal muscle is that the cells start out as a whole lot of little cells. They fuse together during development, and as a consequence they end up with multiple nuclei. So the nuclei are spread out through the cell length, and this is helpful because remember the nuclei control cell activity, and since you have a lot of them, you don't have a real problem with, and since these cells are big, you don't have a problem with things having to wait a long time to diffuse, since you have nuclei throughout the cells. We find this alternating dark and light band. This is the striation, and these are alternating actin and myosin protein filaments, and we'll talk more about that when we get to the muscle lecture. Here we can see a drawing of skeletal muscles and a picture of skeletal muscles, and you can see these alternating dark and light bands. The dark bands are myosin and the light bands are actin, and when the muscle contracts with this orientation, the left and the right sides of this picture would get closer together, and you can see the multiple nuclei in here, right? You can see them there. Smooth muscle tissue, smooth muscle tissue has a couple of functions. One is smooth muscle tissue is capable of creating pressure gradients. We use that in the aorta, for example. When it gets stretched, it contracts against a stretch, and it helps push the blood through the vessel even when the heart is not contracting. It can be involved in thermoregulation as well. This thermoregulation thing is an indirect effect, and we'll see that in the next picture. So here we have something that's not unlike Pansy. We have some red hair and some dark skin, and this would be goosebumps, right? So when Pansy's cold, or her hair stands up on end, and then I didn't draw it in, but these hairs would actually overlap, sort of like shingles, and they would seal off this air underneath them from the air outside. Air is a good thermal insulator. It doesn't conduct thermal energy very well. So this is like wearing a down vest, and it means that the skin won't be cooled off as much as it would be if the hair weren't there. So we do have this goosebump phenomenon happening under two conditions. Pansy knows them. One would be right when she's frightened, and the other would be right when she's cold. Okay, so I'm going to show you a picture of a cat. Pansy's going to really like this because she likes cats. And we're going to see if the cat is frightened or cold. I'm going to let Pansy judge. Right, the cat is frightened. So this is strong sympathetic stimulation of this cat. And you can see a couple of things are going on here. One, the hair is raised on the back, and that's what we were just talking about. And also you can see the cat's eyes are dilated, and that is so it will dilate rather than die early. The advantage of this hair being raised when a cat is afraid is that it makes the cat look bigger, and then maybe the predator that's thinking about attacking this cat will think twice. There actually is a second advantage I hope we'll get to talk about, and that is that by having the hair stand up on end like this, it's called a false target. And so a predator might attack where it thinks the spinal cord and vertebral column are, and instead get a mouthful of hair, in which case the cat would survive and be able to run away. Smooth muscle cells are not really long like the skeletal muscle cells. They're spindle shaped. There are no striations, and they are not under conscious control. They are under control, they're just not under conscious control. Their muscle contractions tend to be slower than skeletal muscle. The good news is that even though they're slow, they can maintain contractions for a long period of time, and they use a lot less energy than skeletal muscles, but they're also not as strong. It's kind of a trade-off. Okay. And then here's a really important characteristic of smooth muscles. Smooth muscles can contract in response to being stretched. This happens in peristalsis in the small intestine particularly, but it also is really important in blood vessels for them to do their job. So if a tube made out of smooth muscles is stretched, it will automatically contract against the stretch. The advantage of this is obviously to reduce the risk that the tube will rupture. If this were not the case, if you didn't have this contraction against being stretched, they would just keep stretching and stretching and stretching until they ruptured, and that would be bad in most cases. So this response to stretching is actually done through self-excitation. In some areas in the body, smooth muscles can undergo self-excitation anyway, and this can be supplemented in terms of control by way of nerves and hormones. Here's a drawing and a picture of smooth muscles. So we see the spindle shape here of the cells in the picture, in this drawing. And you can see the individual single nuclei inside of the cells. Where do we find smooth muscles? Well, mostly in the walls of tubular organs, although not exclusively. So tubular organs, the stomach, the small intestine, the urinary bladder, the uterus, the walls of the aorta. Okay. A second place that we find smooth muscle cells is attached to the base of hairs on our body surface, and these are the muscles that contract and give you goosebumps when you get cold or when you're frightened. Cardiac cells are next, and these cells are going to be cylindrical. They're bigger than smooth muscle, a little bit more similar to skeletal in some ways. They are striated like skeletal muscle cells, but instead of having multiple nuclei like skeletal muscles have, they have single nuclei like smooth muscles have. Their cells are branched, they interact with each other. That's pretty important. And they are involuntary, and this is another characteristic that's shared with the smooth muscles. Smooth muscle cells, remember, are involuntary. Okay. Cardiac cells are kind of similar to skeletal muscle cells in some ways. One of the things that's similar to skeletal muscle cells is that they have strong contractions. They have the striations. I already mentioned that. And then the characteristics similar to smooth muscle cells, they're involuntary. They do have self-excitation just like smooth muscles, so you don't have to have nerves. This is actually really important because when a person has a heart transplant, the heart actually can continue to beat without having the nerves all hooked up. And that's a good thing because typically when you damage nerves or cut them, they don't do real well in terms of recuperating. The activity of the heart can be affected by hormones, and it can be affected by nerves. Primary nerve controlling cardiac activity is the vagus. The vagus nerve is a parasympathetic nerve, and when it stimulates the heart, it actually inhibits the cardiac activity. In fact, strong vagal stimulation will cause your heart to stop. Normally it will start again on its own, but initially it will stop it. Cardiac muscle cells have single nuclei, just like the smooth muscle do. So cardiac muscle is going to be found in the walls of the heart. In terms of function, it creates pressure to pump the blood. Remember that smooth muscles also create pressure gradients. And for that matter, so do skeletal muscles. Cardiac muscle cells also include specialized cells that are referred to as excitatory fibers. And these function very much like a nerve in spreading impulses through the heart so that the heart can contract as a unit. The cells are connected to each other. This is called, this happens by, excuse me, a feature called intercalated discs or gap junctions, and it means that the cardiac muscle tissue can act as a syncytium. This is a fancy term to indicate that they act as one. They act like a team. They all do the same thing at the same time, pretty much. Here are some drawings and pictures of cardiac muscle cells. You can see these places where they join together. Organs that are mostly muscle tissue. I'll give you an organ. You tell me which kind of muscle tissue it is. We'll start with biceps brachia. What kind of muscle tissue is it? Right, skeletal muscle. The heart. What kind of muscle tissue? Right, cardiac. Ooh, the uterus. Oh, she didn't get a chance on that one. Uterus is mostly smooth muscle tissue. The aorta is mostly smooth muscle tissue. And the heart is mostly cardiac muscle tissue. Wow. Nerve tissue is next. Again, there's going to be a separate quiz on nerves and how they work and all that sort of stuff. And nerve tissue functionally is important because it can give rise to or conduct impulses. This is a communication network within our body. Now we communicate from our control centers to our cells and tissues and organs in another way too, besides the nerves, and that would be right, hormones. But let's compare nerve and hormones for a second. What would be an advantage of nerve tissue over hormone? Right, it's very fast. Also, nerves tend to be very well directed. So, if you have a nerve impulse, it doesn't affect all the tissues in your body, or it doesn't come in contact with all the tissues in your body. Whereas hormones that are circulated by the circulatory system have the potential to come in contact with all your cells. In addition to the nerves or neurons, actually nerves are groups of neurons all bundled together. In addition to the cells, the neurons, there are supportive cells called neuroglia, and they do a number of supportive functions to help these cells survive. So there are basically 9 of the supportive cells for each one of the neurons. It's a little bit like a professional sports team and their support staff. So, you take any team that you're interested in and you have the players, the athletes, but then there are all these other people that support them, that make sure that the place where they play their games is kept up, that pay them, that provide uniforms, that clean the facilities, that advertise, that arrange for transportation, all those sorts of things. Those are the supportive people, and the neuroglia are sort of like that as well. So the neurons are going to have 3 parts. I like to use the analogy of a neuron being like a tree. So a tree would have roots and a trunk and then branches, and the roots would be the dendrites. This is where the impulse usually begins that spreads through a neuron. That leads to the trunk. That would be the cell body. And the cell body is the portion of the cell that has most of the organelles. And then we have the axons. And the axons are like the branches of a tree. And I think a really good tree to think of when you think of nerves is a palm tree. So a palm tree has these roots down in the soil, and then a really long trunk. And then at the top of the tree, there are all these branches. And that's kind of what the neurons are usually like. They usually have a whole bunch of connections at the end of the axon. But the axon can be pretty long. Not always, but pretty long most of the time. And these axons are going to react with the next cell. The next cell is going to be after a synapse. Synapse is a space between the cells, and the next cell may be another neuron, or it may be some cell in an organ that is going to be activated or inhibited by the action of the axon. The axons don't actually directly pass their impulses to the next cell. We'll talk about that. That happens by way of chemicals that the axon releases. So here is a model of a neuron, and this would be a palm tree that doesn't have really very many branches at the end at the top. But most axons would have a lot of interactions up here with other cells. So these are the dendrites down here at the bottom. This is where the action potential usually starts, and then it moves up to the cell body and then out through the axon. And then the axon releases chemicals that affect this postsynaptic cell. So neurons are found in basically three places: the brain, the spinal cord, and the nerves. The axons are usually long, and those are called fibers. And in the central nervous system, CNS stands for central nervous system, the fibers come together to form tracts. There are ascending tracts and descending tracts that run through the spinal cord. And then in the peripheral nervous system, the fibers form the nerves. Good thing I didn't have any alcoholic beverages at lunch. Okay, so the neurons are going to be of three types. We have afferent or sensory neurons that bring information into the central nervous system. We have efferent or motor neurons. They take information out from the central nervous system. And then we have the interneurons, and the interneurons are going to exist between the afferent and the efferent in reflexes, and the afferent in the brain, or the brain and the efferent in normal paths. Here's some bad news, and I don't think there's any good news that goes along with it. Neurons don't divide very much at all, if at all, as you age. So, and they're also very poor at repair. So generally, if you damage or kill a nerve cell, you don't get a new one. And that means that impairment of nerves can be, at the very least, they tend to be long-term impairment, and they may be permanent. Another unfortunate thing, and again, I can't think of anything good about this, they have very high requirements for oxygen and for glucose. And what that means is that if you cut the blood supply off to nerve tissue, it tends to die, and it doesn't take long. The neuroglia, remember I told you that they are the supportive cells. There are 4 types of glial cells in the central nervous system, and then there are 2 more in the peripheral nervous system. One of them in the central nervous system, the microglia. These phagocytize debris. If you remember from topic 3, phagocytosis is sort of like Pac-Man. They gobble this junk up, and then typically they metabolize it and break it down and get rid of the waste product. We have astrocytes. They are going to help get nutrients into the neurons. Oligodendrocytes, they form myelin in the central nervous system. There's a comparable cell in the peripheral nervous system called a Schwann cell, and they produce myelin out there. Myelin is a fatty material that insulates the cells. And then ependymal cells, they are going to line the cavities of the brain. Okay. For the peripheral nervous system, two kinds of supportive cells: the Schwann cells and the satellite cells. Okay, the Schwann cells, as I mentioned, create myelin, and this is going to give the nerve, or the bundles of nerves, neurons all together will have this white appearance. And what we find is the myelin is sort of like leg warmers around the axons of the cells. They, in between the myelin, there are going to be breaks called the nodes of Ranvier, and we find that when an impulse moves over an axon that's myelinated, it jumps from node to node, which gives more speed and requires less energy to recover from. This is another case of increasing speed. And then the satellite cells are also out in the peripheral nervous system. Here's the example of how a Schwann cell works. We have the Schwann cell nucleus, and it's creating this myelin sheath that wraps around the axon, as I mentioned, sort of like a leg warmer. And they are in the peripheral nervous system. Okay, we'll do more on Schwann cells and supportive cells and nerves and everything when we get to that quiz topic later in the course. In terms of reproductive tissue, not everybody classifies reproductive tissue as its own type, but I think it's reasonable to do that because the cells are so unusual. So when reproductive cells from two individuals come together, you get a new person. And the cells that come together are called gametes. Okay? And the male gamete is called a spermatozoon. That would be singular. Spermatozoa is plural. And the female gametes are ova. That's plural. Ovum would be singular. So here's a drawing of a spermatozoon. Up here at the top of the drawing is where the genetic material is. And this intermediate piece is where the mitochondria are. The mitochondria is sometimes referred to as fancy nose, right, powerhouse of the cell. And then we have the flagellum. Remember, the flagellum is a non-membranous organelle that propels the cell. So these can swim. They don't start out their life like this. This takes about 20 days of development in the duct of the epididymis in the male. When they start out, they're just a kind of a vanilla cell, but they do differentiate into this highly modified shape. And they do swim. Here's an ovum. Ova do not swim. They're really large. Compared to the spermatozoon, they're huge. They have this outer membrane that has to be digested in order for fertilization to occur. We'll talk more about that later on in the course. Okay. So, extracellular junctions, glands, and membranes, another topic on tissues and membranes. When we have interaction between adjacent cells, this can happen by extracellular junctions, and this helps coordinate how the cells behave. They can act in unison. One kind is tight junctions, another are gap junctions, and then we have adhesion junctions. So tight junctions, we'll start with those. At tight junctions, there's an impermeable barrier, so the proteins of adjacent membranes are tied together, and this just makes the tissue very strong. Okay. In the stomach, there are some of these tight junctions that contain digestive enzymes, and the enzymes are obviously going to break down the food tissue that or the food materials that you've consumed, mostly proteins. In the kidney, the tight junctions actually contain urine. The gap junctions are found in the heart, for example, and there are channels between two adjacent cells so that the cytoplasm is in contact. This is a very strong association. I don't mean strong in terms of strength, but an association that leads to a great amount of coordination between activities. And so things that go through these channels between the cells include ions. We talked about those, sugars, other small molecules. And as I mentioned, These gap junctions are really important, especially in the heart, but also in some smooth muscles, so that we can synchronize contractions so that they occur at the same time. And you might think, well, you know, so what's the big deal? Why is synchronization important? The synchronization of the contractions in the heart increases the efficiency of the heart dramatically. Okay, adhesion junction, desmosomes. Here the membranes are not in contact directly, but they are held together through filaments outside of the cell. There are cytoplasmic portions of the cell, plaques, that the filaments attach to, and this gives strength to the tissue and helps prevent the tissue from tearing. In the heart, that's really important, because if the tissue were to tear, then its effectiveness as a pump would be very much diminished. In the cervix, we have the same kinds of attachment. Obviously, this is smooth muscle here, and this allows the cervix to be stretched but not tear during birth. Cervix is a fairly narrow opening between the body of the uterus and the vagina. So here we have a drawing of an adhesion junction, and so we've got these proteins here. The plaque is like an anchor for those, and then these keratin filaments, those are, remember, proteins that we find a lot of times within these the epithelial tissues. Those are going to be anchored to the plaque as well. So this all leads to stability between the cells so they don't rip. Glands. Most of the glands are going to have cells that are secretory. Usually they're epithelial, and the product that's secreted is usually released by exocytosis. We covered this in quiz 3, right? That's when those vesicles inside the cell move toward the cell membrane, and then they pop, and whatever is inside the vesicle is released into the interstitial fluid. There are also goblet cells. Usually those are columnar epithelial cells, and they secrete mucus onto a surface. Well, at least they usually do. Okay. Exocrine glands are going to have ducts, and the ducts are going to transport the material from the cells to some location. It could be external, like sweat glands, for example, or sebaceous glands in your skin, or they could be internal, like the liver with its hepatic ducts that eventually lead to the common bile duct and deposit their secretions into the duodenum. So sweat glands are going to go to a surface. They have a simple duct. Pancreas secretes into the duodenum cavity, and it has compound ducts. And I have some drawings to show you the difference between simple ducts and compound ducts. Okay. Lactiferous glands have compound ducts. Lactiferous glands produce what, Pansy? Right, milk, and they secrete out to the surface of the nipple. So here are some drawings of types of ducts. We have tubular ducts. This one's obviously very simple, going to the more highly convoluted. Compound ducts with many branches of each, and even the branches have branches, right? And then alveolar structures. The alveolar structure is referencing the same kind of thing that we see within the lung. The alveoli of the lung are sort of globose. They sort of are a spherical cross-section almost. Endocrine glands do not have ducts, and they make their secretions by exocytosis to into the interstitial fluid, typically, and from there the materials diffuse into the blood or into the lymphatic system. They are going to produce hormones, and most of the time it goes into the blood, but it's possible for it to go into the lymphatic system. The lymphatic system eventually drains back to the circulatory system anyway, so ultimately it gets into the blood. And then the blood is pumped through the body and delivers the materials to wherever they're likely to go. So hormones are another way of communicating. We talked about this a little while ago, right? One way of communicating is using nerves, and they are fast. Another way of communicating is using hormones, and they are not fast. And these hormones may affect a single target organ or many target organs or many cells. The drawback, the downside of endocrine secretions is that they tend to be slower than nerve impulses. So, membranes, there are 5 kinds. We have mucous membranes, serous membranes, synovial membranes, the meninges, and cutaneous membranes. We'll talk about some of each of these characteristics. So we can start with the mucous membranes. Mucous membranes are going to be lining the deep walls of organs and tubes. Usually these tubes open to the outside someplace. And the examples here are respiratory, digestive, urinary, and reproductive. The function of the mucus actually varies depending on the system. So in the digestive system, bactericidal and lubrication are the 2 things that are really important, whereas in a respiratory, we warm and moisten the air, filter the air, and bactericidal is helpful. Urinary is mostly lubrication and bactericidal, and then reproductive system, again, lubrication and bactericidal characteristics of the mucus. The epithelium is going to have loose connective tissue deep to it. We have I say that every time, sorry about that. Goblet cells, must be thinking of globulins or something, I'm not really sure. Maybe it's Kepansi's fault. But the goblet cells are in the epithelium and they secrete the mucus. And as I mentioned, mucus is bactericidal and that is beneficial. Also, in the gastrointestinal tract, they tend to buffer the acids a little bit, and that helps protect our tissues. Serous membranes, they occur in a couple of different places. Serous membranes line the cavities. For example, the parietal pleura lines the thoracic cavity, and the parietal peritoneum lines the abdomen. And then they cover internal organs. Typically that's the tissues are lumped into visceral pleura and visceral peritoneum as a generic name, and then you can have specific names for the organs by organ. So the pleura covering the lung would be pulmonary pleura. Okay, so parietal covers the walls and visceral covers the organs. Okay, the serous membranes that are inside these cavities are going to be simple epithelial layers. In fact, when you dissect an animal in the anatomy labs, when we see this epithelium, it looks like saran wrap. You can see right through it. It's clear. Very, very thin, and it takes very little impact to just tear it away. Okay, so simple layer of epithelium covering some loose connective tissue. This serous membranes are going to support organs. So that's more of an important function actually in the abdomen, where the ligaments keep the organs from twisting, especially the intestines. It also, these serous membranes can reduce or slow down the spread of infections if one should occur. They secrete a serous fluid. Now the serous fluid has a lot of different functions depending on where it is. In the thoracic cavity, the serous fluid is important for management of heat, so heat dissipation. It's important for lubrication and for surface tension for breathing. And so we have all of those here. In the abdomen, surface tension is not really a big deal. The other 2 characteristics are still important, though, the other 2 attributes. So lubricating the organs and dissipating heat. Okay. Pleura, I mentioned that already. Going to be found in the thoracic cavity. Parietal pleura lines the walls. We have pulmonary pleura. Pericardium is a type of pleura. It's modified, actually includes some connective tissue to help it be stronger so it doesn't rip. Peritoneum is in the abdomen. And one of the really important ligaments in the abdomen that's made out of peritoneum is called a mesentery or mesenteric ligament. And that is extremely important in preventing your small intestine from tying itself in knots. That would kill you. Synovial membranes, primarily found in joints. And these are unusual membranes because they're not epithelial, they're connective tissue. And they do secrete synovial fluids. And the synovial fluid is going to be really important in lubricating the joints and dissipating heat within the joint. The meninges, these are membranes associated with the central nervous system. There are 3 layers. Pia mater is a vascular layer, the innermost layer that adheres to the surface of the brain and the spinal cord. The arachnoid mater is in between. It's suspended away from the pia mater by the cerebrospinal fluid. Arachnoid suggests it looks like spiders. It doesn't really look like spiders as much as it looks like spider webs. And then the dura mater is a tough outer layer. Typically the arachnoid mater is pushed up against the dura mater, so there's a potential space between them. And then the dura mater is usually pushed up against the bone. So they're almost like 2 layers of a wall covering or something right up against the bone. They're not attached to each other, they're not attached to the bone, but they are pressed together because of the cerebrospinal fluid. These are found in the posterior cavity. Remember, the posterior cavity includes the cranium and the vertebral cavity. They are connective tissue. I mentioned that, I think, already. And they're going to protect the brain and the spinal cord. Sometimes students come to believe that they are part of the brain and the spinal cord. They are not. They are covering. Just like if you wear a shirt, the shirt is not part of your skin. It's a covering of your skin. So these are coverings. And then we have the cutaneous membranes. The skin, there's going to be an entire quiz topic coming up on the skin, actually. This is the outer covering of our body, and primarily it's going to be epithelial tissue. You'll recall that we talked about keratinized stratified squamous cells. Those help the skin act as a barrier. And then there's some dense irregular connective tissue deep to the epithelial tissue. So that gets us through quiz 4 on bodies and the body's tissues and membranes. And Pansy has gone into a coma. She's become so tired. I hope you enjoyed this and got some useful information from it. And I'll be working on a possible answer list for this so that you can prepare yourselves for the quiz associated with this. And this material will be in the second exam. I hope you have a pleasant evening.